Today, we're going to decode one of the most fundamental processes in all of biology. We're talking about how your cells read the instructions in your DNA to build, well, everything that makes you, you.
So the central puzzle of genetics really starts with a numbers game. How does life build all of its incredible complexity from a language that has only 4 letters?
Yep, just 4. When that genetic recipe is carried by messenger RNA, the entire alphabet consists of adenine, uracil, guanine, and cytosine. A, U, G, and C. That's it. Just 4 characters to build all of life as we know it.
But here's the thing. Those simple instructions have a huge job. They have to build the complex machinery of our bodies. And that machinery is made of proteins. These are the workers, the enzymes, the very structure of our cells.
And get this, proteins are built from a set of 20 different amino acids. So, here's the big question. How do only 4 bases spell out the instructions for 20 different amino acids? It really doesn't seem like we have enough letters in the alphabet. So here's how we're going to break it all down. We'll start with how scientists cracked that triplet code, then we'll get into the rules of this genetic language, we'll meet the special translator molecule, visit the protein factory itself, and then wrap it all up with a final recap.
You know, back in the mid-20th century, scientists were scratching their heads over this exact problem. And they solved it not just with experiments in the lab, but with pure logic and a little bit of math.
They started thinking it through. Okay, a 1-letter code, where 1 base equals 1 amino acid, that only gives you 4 combinations. Not nearly enough. All right, what about a 2-letter code, like AG or UC? Well, that's 4 squared, which gives you 16 combinations. We're getting closer, but still not enough to code for all 20 amino acids.
The big breakthrough actually came from a physicist, of all people, George Gamow. He proposed a wild idea. What if the cell doesn't read letters one by one, but in groups of 3? He called them codons.
And suddenly, the math just clicks. 4 cubed, that's 4 times 4 times 4, gives you 64 possible combinations. That is more than enough to specify all 20 amino acids, and you even have some left over. This was the huge aha moment.
So, this is the absolute key takeaway. The genetic code isn't read letter by letter, it's read in 3-lettered words that we call codons. Every single set of 3 bases on a messenger RNA molecule is 1 word in a long genetic sentence.
And here it is, the complete solution. This codon table is basically the Rosetta Stone of biology. It's a dictionary that shows you exactly how each of those 64 3-letter codons translates to a specific amino acid. For instance, if you look up CUU, you'll see it codes for the amino acid leucine. It's all right there.
Okay, so we've got the words, but you know, any good language also needs grammar. And it turns out, this genetic language has a very specific set of rules.
So here are the key features. First, the code is degenerate, which is just a fancy way of saying it has some redundancy. You can see that 4 different codons all code for proline, but, and this is super important, it's also unambiguous. Any single codon will only ever code for 1 specific amino acid. No confusion there. What's truly mind-blowing is that it's almost universal across all life on Earth. And finally, there's no punctuation. It's read continuously, like a long sentence with no spaces, from a very clear start signal to one of 3 stop signals.
Because the code is read in these continuous sets of 3, staying in the right reading frame is absolutely critical. Think about this simple sentence made of 3-letter words.
Now watch what happens if we just insert 1 extra letter, a B. The first word is still fine, but look at everything after it. The entire sentence gets scrambled into complete gibberish. The meaning is totally lost.
This is what we call a frameshift mutation, and in a cell, it's often catastrophic for the final protein. It just perfectly highlights how incredibly precise this whole reading process has to be. So, if the messenger RNA has all the codon words, what molecule is actually doing the reading and translating? How does the cell connect the genetic word to the right amino acid? There has to be a translator, right?
Well, meet the star of the show, transfer RNA, or tRNA for short.
This little molecule is the master translator. And what's so incredible is its unique structure. It's a single strand of RNA that actually folds back on itself into this distinctive cloverleaf shape. It's literally built for the job.
And I say it's built for the job because this molecule is essentially bilingual.
On one end, it has what's called an anti-codon loop, with 3 bases that are the perfect complement to an mRNA codon. Then on the other end, it carries the one specific amino acid that corresponds to that codon. You see, it's the physical bridge that connects the two languages.
Okay, so we've got our mRNA message, we've got our tRNA translator, and we've got the amino acids. Now, let's head to the factory floor, where all of these components come together to actually build a protein. This whole process is called translation, and it happens on this massive molecular machine called the ribosome. The ribosome literally clamps onto the mRNA strand and acts as a kind of workbench where the protein gets built.
The process unfolds in 3 main stages. First, in initiation, the ribosome finds the AUG start codon and gets everything set up. Then, during elongation, the ribosome starts chugging along the mRNA, one codon at a time. The correct tRNA docks, delivers its amino acid, and then leaves. The ribosome links the amino acids together, and the protein chain gets longer and longer. Finally, when the ribosome hits a stop codon, that signals termination. The whole assembly comes apart, and the brand new protein is released, ready to go to work.
Okay, so let's put it all together and do one final recap of this absolutely elegant system.
The core logic is the triplet code, which solves that 4-base to 20 amino acid problem with that simple formula, 4 cubed equals 64. The mRNA acts as the message, which is read in 3 base words called codons. The tRNA is our crucial translator, and the entire operation takes place inside the ribosome, the protein synthesis factory.
So I'll leave you with this final thought. This complex, incredible system is virtually the same in you, in a mushroom, in a tree, and in the bacteria in your gut. This shared language of life is one of the most powerful pieces of evidence we have for a single common ancestor for all life on Earth. Pretty amazing, isn't it?